ஹலோ ஹலோ சார் கேட்குதா? சார் எனக்கு யூடியூப் நாங்கள் லைவ் கான்ஃபரன்ஸ் போயிட்டுருக்கு சார். எனக்கு லேண்ட் மட்டும் என்னோடய லேப்டாப்பில் கனெக்ட் பண்ணோம் சார். நான் எங்கள் ஹெச்ஓடி ரூம் கேபினில் தான் இருக்கேன். லேண்ட் பக்கத்தில் இருக்குது. அதுக்கு நம்ம லேப்டாப்பில் கனெக்ட் பண்ண மட்டும் வந்து பண்ணி கொடுங்க.
நீங்கள் டெஸ்ட் பண்ணாலும் லிங்க் பண்ணுங்க.
ஓகே சார்.
சரி அம்சேக்கிறேன் மேடம். ஓகே ஓகே மேடம். ஆல்மோஸ்ட் மேடம். அது அனுப்புகிறேன் உடனே மேடம். பிபி மேடம்க்கு ஏதாவது லிங்க் வரலையாமே?
லிங்க் கேட்டாங்க சார். இப்போ தான்
லிங்க் சொல்லு. இது எதுக்கு இது இதெல்லாம்
சார்? நெட்டு லைவில் கனெக்ட் ஆக மாட்டேங்குது. நம்மளுடைய நெட்டு எரர் வருது, டேட்டா இல்லைன்னு.
இது ரொம்ப மஸ்ட்டாக இருக்குது இந்த இன்டர்நெட்டு. இது உங்களுக்கு கண்டிப்பாக வேணும் உனக்கு. சரி இப்போது இந்த லிங்க் சொல்லு.
ஸ்வாதி சுதாகர் ஜாயின் ஃபைனல்ஸ்
மாட்டாங்களா?
ஃபைனல்ஸா?
எக்ஸாக்ட்லி 5 minutes எடுக்குமா? 5 minutes 5 seconds 15 seconds கொஞ்சம் வீடியோ 5 minutesனா வேற. 5 minutesனா நான் பேசணும்.
லிங்க் பண்ணி ஷேர் பண்ணிடுங்க.
Now I request our department head, Mr. Hem
College Chennai, for joining us today to share their expertise and knowledge with us. We extend our sincere thanks to our institutional leadership, respected Chairman Dr. K.P. Gautam Srinivasa, Managing Director Dr. K.P. Arvind Srinivasa, Principal Dr. K. Somasundaram Sir, Vice Principal Dr. V. Anita Ma'am, and the heads of the organizing departments for their vision, support, and continuous encouragement throughout the planning and execution of this conference. We thank all the department heads and faculties for their support. We also express our sincere appreciation to our key experts and session chairs who have taken time out of their busy schedules to share their expertise and knowledge with us. Your contributions are what make this international platform meaningful and impactful. We deeply appreciate the effort of our organizing committee members, coordinators, and technical support teams who have worked tirelessly to ensure the smooth conduct of this online event. To all our participants and paper presenters, thank you for your active involvement and valuable contributions. Your research and discussions will be the heart of this conference, and we look forward to a series of engaging and productive paper presentations. Lastly, I thank each and every one of you who joined us virtually today. Your presence has made us this inaugural session a memorable success. Thank you all once again.
Thank you, ma'am. Now I request all the participants to leave the section. The next section will be started at 10:15 by Swati Solakar, ma'am. Thank you. The preliminary talk section link will be shared in the WhatsApp group. Please do join in that.
இவனை ஏமா வர வச்சிருக்கேன் இந்த ஆண்டு வந்து மோடல் பண்ணாலும் பரவாயில்ல. ஏதாவது ப்ரோக்ராம் என்ன இதை கனெக்ட் பண்ணி விடுறேன். இதை கனெக்ஷன் கட் பண்ணி விடுறேன். அதுல இருந்தா நான் இங்க கனெக்ட் பண்ணி வச்சிருந்தேன். எல்லாம் போச்சு பாருங்க.
அங்க லைவே போயிட்டு இருக்கு.
இல்ல இல்ல இவனை ஏன் வந்து அந்த ஈத்தர்நெட் கேபிளையும் எடுத்தான்?
கேபிள் நமக்கு கிடையாதுன்னா அதுதான் வைஃபைல கனெக்ட் பண்ணி
அதை தான் தூக்கி கனெக்ட் பண்ணி வச்சிருக்கானா? 2 நிமிஷத்துல டக்குன்னு ஆயிடுச்சு இந்த விஷயம். சரி எங்க சொல்லி கொடுத்தோமா?
செக் செக் பண்ணா ஐ சி எவ்ரிடே
என்னது?
லைவ் வரதா? இது கான்ஃபரன்ஸ்
லைவ் வருதா இல்லையான்னு நீ தானே பார்த்து சொல்லணும்.
எனக்கு இங்க ஸ்ட்ரீமிங் ஆகுது சார். நான் வந்து கான்ஃபரன்ஸ் எஸ்எம்ஐடி போட்டு வருதான்னு கேக்குறேன். நான் கான்ஃபரன்ஸ்
யூடியூப்ல பார்க்கலாம்.
ஆமா அண்டர்ஸ்கோர் பயோ அண்டர்ஸ்கோர்
பிரபஞ்சா உன் ஃபோன் எங்க இருக்கு சிக்னல் நல்லா?
சார் எனக்கு எல்லாமே சொல்லுங்க.
சரி ஓகே. இப்போ அங்கே உட்காந்து நீங்க ஃபோன்லயே பண்ணுங்க. காம்பேரிங் மட்டும் தானே பண்ண போறீங்க. யூ டோன்ட் நீட் a laptop. ஓகே யூ ஜஸ்ட் நீட் a phone.
சார் ஏதாவது ஒரு இதுக்கு வந்துச்சுன்னா ஏதாவது ஒரு சின்ன செகண்ட்
சிக்னல் இல்லைன்றியே அதுதான் பயப்படும். அந்த அந்த சைடு விண்டோ ஓப்பன் பண்ணுங்க. விண்டோ ஓப்பன் பண்ணா அதெல்லாம் ரிக்வெஸ்ட் பண்ணுங்க.
ஸ்மால் கரெக்ஷன்ஸ்க்கு நம்ம வந்து
சரி சரி ஓகே ஓகே. நோ ப்ராப்ளம் நோ ப்ராப்ளம். இங்கே பாருங்க. சரி இப்போ அவங்களோட பிபிடி வாங்கிடலாமா? வாங்கிட்டு அவங்களே
திடீர்னு ஸ்டாப் ஆயிடுச்சு. சரி ஸ்டக் ஆயிடுச்சு.
பேசலாமா?
சரி கேட்கல. லைவ்ல ஹலோ
அவங்க ப்ரெசென்ட் பண்ணிக்கிறாங்களா? அப்படி வரலன்னா நான் உங்களுக்கு பிபிடி அனுப்புறேன், நீங்க ஷேர் பண்ணுங்க சார்னு சொல்றாங்க ஸ்க்ரீனு. ஓகேவா? ஸோ அவங்களுடைய ப்ரொஃபைல் ஃபர்ஸ்ட் நம்ம பேசணும். இப்போ அவங்க கிட்ட கேட்டால் செருப்பு அடி விடுவோம்.
கூகுள்ல போட்டு எடுத்துக்கலாம் சார்.
இருக்கு
சார்
ஃபர்ஸ்ட் நீங்க ஸ்டார்ட் பண்ணும்போது குட் மார்னிங், வெல்கம் பேக் டு ஆல் த பார்ட்டிசிபென்ட்ஸ். நவ் இட்ஸ் டைம் ஃபார் தி கான்ஃபரன்ஸ் plenary talk session. Now I request head of the department, Swati Sri Madam from AP America to give away the chief guest profile questions. தரேன் ஏன் வேணும்
சார் கூகுளே வந்துருச்சு சார்.
அப்படியா? அப்படின்னா இருக்கிறது அப்படியே படி.
டைம் நீங்க பேசுறது கேட்குது.
Society for Applied Mechanics, member of Indian Society. And biophysics. So with these warm
ஆல்வேஸ் அந்த மாதிரி இப்போ இந்த ஆரம்பிச்சா மட்டும்தான் நீங்கள் போகணும் மேடம் அவர் 9 to 3:30 ஆனால் அது வந்து ஆக்சுவலாக பாருங்கள் அவங்களோட ட்ரெஸ் கோட்லேருந்து எல்லாமே இந்த ரிவ்யூ ஹைலி ப்ரொஃபஷனல் ஓகேங்களா இப்போ வேரிங் ஓவர் ஒன்லி டே இருக்காங்க MBAனாலே இங்கே தனியாக தெரிவாங்க ரிவ்யூ எல்லாம் அதே மாதிரி மோட்டர் பேஸ்மெண்ட் பற்றி நீங்கள் கவலைப்படாது அதே மாதிரி எக்ஸலன்ட் ரிசல்ட்ஸும்
ஓகே என்னன்னா ஃபஸ்ட்டு அவங்க பேசுகிறது பேக் எண்ட்
நீங்கள் தனியாக ஒரு இடத்துல வச்சு பாருங்க நீங்கள் தான் லைஃப் பண்ணுறீங்க
நீங்கள் ஏற்கனவே பார்த்துட்டு
சரி நாளைக்கு எங்கள் கேபின்
எங்கேயோ தனியாக எங்கே போய் சொல்லி நான் சொல்லி சரி
நான் உங்களுக்கு கால் பண்ணேன் இங்கே பிஸி இல்லை சரி அதனால் நான் சொன்னேன் 5 minutes பிரேக் இருக்குல்ல அந்த கேட்டுடுறேன் ஆனால் அவர் பிஸியாக இருப்பார் எனக்கு வந்து லைவ் போயிடுச்சு லைவில் நம்ம பேசுகிறது
இல்லை வந்து ஃபஸ்ட் கம் ஃபஸ்ட் சர்வ் பேசுகிறாங்க
அதில் எனக்கு ஒரு விஷயம் இல்லை அப்படி சொல்லுங்கள் தேங்க்யூ
microns. And whatever it is going less than that, it falls under the nanomaterials. It is called 10 to the power minus 9. And there are people started to work on nanoparticles because of their high surface to volume ratios. And it has the cells, how we have a preference to eat. Our cells have a preference to uptake nanomaterials than that of the free drug alone. This is why people started to work on nanomaterials and nanotherapeutics. So here, they started to work on different kinds of nanomaterials like liposomes, dendrimers, hydrogels, carbon nanodots, and polymeric materials.
கான்ஃபரன்ஸ் போயிட்டு இருக்கு, அதனால தான் நான் வந்து பேசிட்டு இருந்தேன். நான் பார்த்துருக்கேன், ரிசப்ஷன் இருக்காங்களா? சரி, எத்தனை பேர் சொல்லுங்க, கீழே இருக்குதா? சரி சார், ஓகே சார், சரி சார்.
What I would say is, let's say we have a gold nanoparticle and silver nanoparticles, and they are they can cure some parts of the diseases. However, if their concentration exceeded some limit, then it induces a toxicity, and also it is not biocompatible in nature. So for them, there is also a lot of limitations, including you can't load that much of drug and drug release efficiency. And the cost efficacies, and these are all the limitations which are associated with the current delivery systems. So first of all, delivery system means, let's say you are there in your college, and if you want to come to IIT Madras, what do you do? Either you can walk or you can take the bus and you can come here. So bus is the delivery vehicle. In the same way, you can load your nanomaterial or drug inside this inside this delivery vehicle. And this nanoparticles, this delivery vehicle can easily transfer the drugs inside the cells. So that is what called as a delivery system. Okay. And so you can see there is a need for next generation materials. So this is what our lab works on. So our lab works on nano archosomes, nano needles, hematite based nanoparticles, and protein nanocages and nanofibers. And we work also on so I don't know whether you people know about liposomes. Have you heard about the liposomes? You should tell yes or no. So then only I can proceed with the nanoarchosomes. Have you heard about the liposomes? So should I take it as yes or no? Okay, I consider it as a no, and then maybe it's better you people also instead of me monotonously talking, if you people also interacting with me, it will be so fun for me also to talk. Don't be get afraid of anything, okay? I'm also a human being as like you people, okay? Instead of making it a monotonous, let's make it as an interactive session. So if you know, you tell yes. If you don't know, you can tell no. So do you know about the liposomes?
No, ma'am.
No, okay. So good. So for example, let's say the cells, right? Our cells are made up of our cells have cell membrane, and the cell membranes are made up of lipids. And this lipids you can take and you can reconstitute them to form a liposomes. And this is this will be in some mode in the nanometer size. And currently if you look under the FDA approved drugs, right? So for the cancer, so these are the liposome loaded doxorubicin, liposome loaded cisplatin. These are the FDA approved drugs which are currently available in the market. So if I want to tell it in layman language, you take the idli, okay, you dip it in the sambar. Okay, sambar is your drug, idli is your vehicle. What will happen when you take the idli and you dip it in the sambar? The sambar will get absorbed into this idli, right? So then that idli you are selling to some people as a sambar idli. So it's the same thing. like idli is here, like, it consider that idli in a nano level. So that is called as liposomes, which are made with lipids. And like, the one which you're dipping it in sambar, right? That is your drug. Okay. So the drug which you're loading in is liposomes and you're selling it as a drug, where like, when you eat the sambar idli, like, what you will get the pleasure and like, you feel energetic, right? In the same way, when like, the things which I was showing, like, these are variety of next generation nanomaterials which we were working on. And then, so this is what I was telling, like, from the hot springs, we can create, like, derive this archaea, and we can create this, this nanoorganisms high temperature, high acid tolerance, and high melting temperature. And so you can see the 10 images, right? Okay. First of all, like, you people know what is transmission electron microscopy? So why we need that term? Have you heard about this term, like, microscopy and transmission electron microscopy? Again, yes or no?
No, ma'am, we don't.
No, ma'am.
Okay, okay. So you are in first year or which year, pa? You people all are from?
I am from second year.
You are from second year. Okay, okay. So let me explain, like, what is transmission electron microscopy. Okay. So let's say, like, okay. So can you see these two objects? Like when I'm just placing it like this, can you see them both?
Yes, ma'am.
Yeah, you can see that, right? Okay. Let's say now, like, can you see people, like, what it is there inside this, like, box?
I don't know. It's like a white thing
So
inside a glass.
you can see what it is, right?
Yes,
So
ma'am.
something is there, like, you can see that. And then, like, there is, I mean, like, in the same way, okay, well, now I will just show you something. Okay. Okay, now. So can you see, like, anything is there inside that?
No, ma'am.
No, right? See, like, we also have a nanoparticles inside that, okay? But we can't see with our normal eyes, okay? So we need microscope. Have you people heard about microscope? Have you used that microscope?
Yes, ma'am.
Okay, you people have used the microscope, okay? So, like, in a microscope, like, we can able to see the objects which are in micron size. But like, whatever it is in nano size, and again, I will say that is a sambar, okay? Okay, what we are doing, we are encapsulating this drug inside this nanoarchosomes, and we are putting them inside this cancer cells, and we are studying how well our teeny idlis can kill the cancer cells. This is what we are doing. Okay, any doubt in this concept? Because based on this only, whatever I'm telling, it will you people can. So what we studied is that, after, you take this idli and dip it in the sambar, you just keep the idli outside on the plate. What will happen? The sambar will start to ooze out, right? The same way, you have a liposomes, which is loaded with the drugs. And when you place them inside our blood, okay, when you're injecting them inside your blood, what will happen is the drugs start to get slowly released from your, this nanoarchosomes, and it will enter into the cancer cells. So that we have studied, and we found one more thing, interesting thing about cancer is that the cancer cells have a acidic pH. Means we are neutral, normal people have a neutral pH blood, and the acidic people, cancer people have an acidic condition of the blood. So it is always better if you, as an engineer, if you create a particles, which can release the drug in a faster fashion. So that is what we did, and we found at acidic pH, our drugs are being get released at a higher fashion. And after that, one thing about this nanoparticle is that they should be biocompatible. What do you mean by biocompatible? Means, they should go and attack only the villain. They should not go and attack the hero, right? So in the same way, whatever you are developing, they should go and attack the Prakash Raj, not the Vijay. Okay. So they should go and kill only the cancer cells, but they should not go and kill the normal cells. So how we know, our, whatever we developed, it is not going and attacking the Vijay, but only the Prakash Raj. So we do this assay, which we call them as a biocompatibility assay. Okay. So what you are seeing, right? This one, and this is the microscopic images of our fibroblast cells. So fibroblast cells, again, I will tell it is a skin cells. Okay. So here you can see this is the control, means we are not treating them with any drug. And this is one which is treated with our formulation which we developed. And this is the drug loaded. The idli dipped in the sambar. So this is the drug loaded nanoarchosomes. And you can see this green color thing, right? So green means the cells all are happy, they are viable. And if you see the red dots means they have died. So what we wanna tell is that the formulation which we have created, the nanoarchosome loaded with the doxorubicin, again in a layman, like an idli loaded with the sambar, it is not affecting, it is not attacking the Vijay, it is not only attacking the Prakash Raj, it is attacking the cancer cells but not attacking the normal cells. So in this way, we have overcome the first drawback of the conventional chemotherapy. Because in chemotherapy, they are attacking, the current conventional chemotherapy, what people are taking currently, that chemotherapy is affecting both the normal cells as well as cancer cells. This is why, they lost their hair and they are becoming weak and all those things. So now we have solved this problem. Our formulation is going to help Vijay, not the Prakash Raj. So next we need to know, let's say, we have created some helper people, right? How it is going to attack this villain. So that is what we need to know about it. So that is what we have done. So what we have done is we took the doxorubicin and we loaded them with the nanoarchosomes. And we, after, we took this, nanoarchosome loaded dogs, we studied how well they could able to, kill the cancer cells. So that is what we did. So we varied the concentration of this, drug loaded doxorubicin, and we studied, how efficiently they can kill the cancer cells. So here you can see when we vary the concentration and when we see the cell viability, with increasing concentration, the cells are dying. So means, whatever you have developed, they are going and killing the Prakash Raj. Even you don't need 100 people or 200 people. with even 6 people, they can efficiently kill the cancer cells. And so what you're seeing, right? This is the microscopic images of the cancer cells. And like, this is the doxorubicin alone, means like, just the free drug. And these are the doxorubicin which are loaded inside this nanoarchosomes. And you can see, like, you can, I mean, this is, maybe I don't know whether you people can see that efficiently. See, like, this is how the cancer cells looks like. And after we treated them with the drug, you can see, like, their structures are, like, disintegrated. It's like, Gilly movie's final climax. So how Prakash Raj has been, like, destroyed completely, right? In the same way, this cancer cells are died completely. And, but you don't need, like, so many people. Like, you don't need 500 nanomolar. But even in 6 nanomolar, you can kill the cells efficiently. Okay. So clear with this concept? Can I move to the next part? Yeah, I mean,
Yes, ma'am.
Okay, great, nice. So you see, like, again, you can see, so these are the live cells which are green, and like, these are the cancer cells which has been dyed. So that is why they are red in color. Okay, so this is what we have found. And like, we have done like a lot more into genetic level, which I don't want to go ahead. And like, this paper has been published in RSC Nanoscale, and like, the cover letter has also been published. And like, we asked another question, like, can nanoarchosomes replace liposomes? Means, there are 2 things. One is oil, one is water. So in the previous study, we have showed that you can encapsulate the oil, water, and it has a better efficacy. Then we have a question that, can we encapsulate also oil-like substances? Means there are different kinds of drugs which are hydrophilic and hydrophobic in nature. So we asked the question, can we encapsulate the hydrophobic molecules also? So that is what we did. So we created the nanoarchosomes. We took the other drug, which is quercetin. Again, in layman language, if I want to tell, it is a manjal. You people might have like heard about the curcumins, right? So it is like compound derived from the turmeric, which we call them as a quercetin. And we encapsulated them, and we also studied their biocompatibilities. And in the same way, how we see the effect in the previous one, it also showed the same effect. They could able to kill the cancer cells efficiently. So this is what, like, in my studies, like, more efficiently we are working on, like, how it is killing the cancer cells, and like, more into molecular pathways, in which way they're killing and all those things. And in addition to that, in our lab, we also work on NIR based, like, the drug release. Means again, like, what it means, like, you have a idli loaded with sambar. Okay. But I want, like, this idli to be work efficiently. So when you pass the light, what will happen is this idli will be get disrupted. The nanoorganism will be get disrupted, and they can release the drug in a faster manner, so that we can kill the cancer cells in, like, in a more efficiently and more higher ways. And this is what the other work which we did, and like, also that we study, like, how well, like, we induce cancer in the zebrafish, and we give our formulation and check, like, how well it can able to kill the cancers in zebrafish also. And the other thing which we also, like, work in our lab is the chick embryo model. So, most of the non-vegetarian people, they might have, like, ate the egg. So, egg, if you leave it for some time, it will be converted into chick, right? So, in the same way, like, this catapas the immune cells to only kill the cancer cells, not the normal cells. And we will also, what we do, we open the gate, like, where the in cancer, like, this all cells has been, like, they are, like, locked up in the chain. So we send some people, like, that is our hero, where is the nanoorganism loaded with the tumor protein. So this, they can go and, like, release the lock where this, all this, like, this people, DC cells, macrophages and T cells are catapas. Okay. So we will release all these cells, so they can efficiently go and kill this cancer cells. Okay. So this is what, like, we do. And so for this, we have created this nano formulations. And then, so we are like, training this, the cells, like, for example, this lymphocyte subtypes. And this is how this catapas looks like, this dendritic cells and macrophages. If you put them under the microscope, this is how they look like. So we collected the cells, and we treat them with the cancer cells. So what you're seeing is the cancer cells, and the bigger one is the Katappa, the smaller one is the cancer cells. So this is how they interact, and they will kill the cancer cells. So as a take home message, what we want to tell is our formulations are are happy, or really good, and evoking the immune system, and this immune cells can go and attack the cancer cells. So this is what other kind of work which we are doing, and whatever work which I have discussed with you, all things are we have filed a patent and patents has been granted. And this one I'll skip because it is too biology, and I'll just show you interesting stuff. Little bit out of the topic. So yeah, everyone it is little bit. Even as a student, I am so interested. See, space biology and space therapeutics is the another sector which we are working along with Indian Space Research Organization. And do you know, Sunita Williams, she went and she was stuck there for so many months, and she came back. While she came back, she can't walk, and she has a problem in speaking and all those things. So have you people think about why so? Why astronauts, when they're came back to Earth, why they couldn't able to move properly, or they're feeling so sick in our Earth condition? Have you people think about that? The answers could be wrong, but have you people had thought about it? Yes or no? Okay, I'll tell. Okay, so what is happening for the astronaut is that we should be thankful to our gravity condition, because while we are walking, there is some force is acting on us, and that force is keeping our whole body in a correct manner. But what will happen is that when you are in space, it is called microgravity condition. Means it is called free floating conditions. Means let's say a skydiver is jumping from the airplane, right? So they will be always in free falling condition. So that condition is called as microgravity condition. Okay. So, in microgravity conditions, what will happen is that, let's say if someone scolds us, we will be get stressed. Let's say mom is scolding us, we will be like, how come she can scold us? Okay, we will be get stressed, right? So in the same way, when our body is in microgravity condition, this microgravity condition will induce us a RO species, means reactive oxygen species. And this reactive oxygen species can go and they affect the our whole body system. So in the same way, if we have a bone, our bone has some kind of cells which we call them as a bone cells. So because of the stress molecule, our bone cells, everything will start to die. And just because of that, the astronauts who are traveling in the space, they will lose their bone. And bone loss, whatever they are having, is around 2 to 8% in a year. So this is what they are having. So this is the problem in astronauts. So in our lab, we created some kind of a formulation, and this formulation, we have checked the stability, and we found that this formulation can able to efficiently, they can able to arrest the RO species, means they can able to arrest the stress molecules which are created in this space. And so yeah, we have done different kinds of tests, different kinds of gene studies. And we found that our formulation can able to reduce the stress as well as induce the osteogenic genes, means the genes responsible for the bone formation. So this we have patented it again, and we are planning to test this formulation in space by developing the payload. That is what we are doing currently. And so yeah, in addition to that, in our lab we work on different kinds of materials like micromotors, so which can propel themselves and they can deliver the drug efficiently inside the cells. And so yeah, we are working on different kinds of films protein and pH responsive films for the wound healing applications. So let's say you have a wound the diabetic ulcer wound and normal wound, you will put the bandage and plaster, but they are not more effective. So what we have done is we created a novel protein-based fibers from the waste, and we found that they could Yeah, so they could efficiently induce the wound healing activity that we have checked in the chick embryo model and also in mice models. So in addition to that, we do like a lot of single molecule biophysics work. So which I just like skip it for the time's sake. And so these are the disease modeling studies which we are doing. Like we are using the zebrafish models and chick embryo models. And in addition to that, like these are the international collaborations which we have all over the world. And these are the lab facilities which I created in IIT Madras. And we have zebrafish, chick embryo, cell culture facilities, and all those things. So yeah, again, like we work on cancer for diagnosis, therapy, prevention, and to prevent also the cancer recurrence. And so like with this, I would like to acknowledge my students and as well as thank IIT Madras and also all my funding bodies. And yeah, I would like to thank the HOD, head of the department, and principal to invite me to present this talk. And the floor is open for the questions. You can, like, let's say let's keep next 5 to 10 minutes for the question and answer section. So I would like to make it an interactive section. Thank you, everyone. So guys, I can yeah. So maybe you can turn on your video.
Session. We'll start with the questionnaire session because actually they are sitting in a classroom setup.
Okay.
So the live streaming is going on. So now the students to interact. Students, you might ask your questions. If they are not audible, you can put the questions in the chat box so that we will read up for the speakers.
So, is it possible to see the students? Can, like if there is some setup, can you please turn on the video so that I can see them? Okay, nice. Okay, guys, come on. What you people understand? You can ask me question because I don't know whether I will also have a time individually. So the make use of this opportunity. If you have any questions, please ask them. It could be of some don't think about the question. If you have something in your brain, ask them.
Is possible to use the osteogenesis medicine you told before to osteoporosis, the old ladies here?
Yeah. So whatever we have developed is not only for astronauts, it can be also used in earth condition. If it is that we are also doing, we also induce the osteoporosis in the fish and we gave a formulation and it is doing very well in treating the osteoporosis also. Yes, Kishant, we can do that.
Thank you, ma'am.
Yeah, maybe you can show your face. I also need to look at Kishant. Okay, thank you, Kishant.
Thank you.
Ma'am, as you said, nanomaterials can treat, can be used for drug delivery, but can it possibly stop the recurrence of cancer?
Yes, that is what we are also working on it. So for example, I didn't present that work. We have created some films. So what after the surgery, one can implant this film and suture it back. So this film is an intelligent film. So only when there is a cancer occurring again, it will release the drug and they will kill the cancer at the early stage itself. So this is also the other work which we are working on it to prevent the cancer recurrence.
Is it very efficient for malignant tumor?
Yeah. So in malignant tumor, let's say the tumor metastasis will happen. So we also proved that it can prevent the metastasis process. However, whatever we proved, it is in chick embryo model. So the next one we need to prove it in rat model and then we need to go to the patient thing. That is what we are progressing towards.
Okay, ma'am.
Thank you, Kirtana. You can also show your face. Okay, thank you, Kirtana. Yeah.
Student.
Ma'am, can we use nanoparticles injected stem cells to cure cancer?
Sorry, can we use nano?
Nanoparticle injected stem cells to cure cancer?
So nanoparticle injected to cure cancer. Is it right? The question is.
Stem cells.
So can we? Okay. So sorry, I couldn't hear it very well. Can you come again?
Ma'am, can we use nanoparticle injected stem cells to cure cancer?
Okay. So nanoparticles injected stem cells. So stem cells is for example, let's say if you lost some portion, okay, stem cells is to regeneration. For the cancer, you need to kill the cancer cells, right? So so the other thing you can do, for example, you can take the nanoparticle loaded with the drug and you can kill the cancer stem cells, but we can't use the stem cells to kill the cancer cells. That is something people are trying to do that. I'm not sure how successful they are. But however, you can use the nanoparticles to kill the cancer stem cells, because even though you kill the cancer, there is something called cancer stem cells. If they are there, there will be again and again the cancer will be coming again. So the main thing we should work on is that not only killing the cancer cells, also killing the cancer stem cells.
Okay, ma'am. Thank you, ma'am.
Yeah.
Okay.
So cool. So if there is not more question, then I would wish you all great success, and I hope you people will have a great conference today. Yeah, thank you all. Thank you, Professor.
Thank you, ma'am. Thank you for your wonderful session, ma'am. If any doubts are being posted, I will give you
Sure.
for any further questions.
Yeah.
Thank you, ma'am.
Thank you.
Thanks. Thank you.
See you
ma'am.
all. Bye, guys.
Cas13 is not used in genome editing. And also a topic called revival of the loss, where the exact process of de-extinction is explained by a biotech company called Colossal Biosciences has successfully done the Diarolf de-extinction process, and we're going to explain the mechanism about it and the bioethical and social implications behind the challenges, what are the ethical concerns behind it, and arising debate or the scientists act like playing God, or it's actually a restoration of ecosystem process. What's the public perception of the de-extinction, and future emerging projects such as the de-extinction process of woolly mammoth. So starting with the introduction. So what is actually, sir, is my slide changing?
No, your slide is not changing, Kailash.
So now it's audible?
Now it is fine. It is changing now.
So starting with the introduction, what is actually de-extinction? It is the process of generating an organism that resembles and is genetically similar to the extinct species, and by resurrecting its lost lineage of core genes, genetically engineering its natural existence, its own adaptability that will allow it to thrive in today's environment and to add a new feature to the existing ecosystem. This term became common among many nations' biotech companies, as it first gained this term in March 2013 in a live stream show called TED Cross De-Extinction, done by a non-profit organization hosted by National Geographic. Actually, it was introduced as a means of undo of historic extinctions by modern tools and techniques such as CRISPR, and restoring the new versions of the extinct species. Not the exact one, but slight modifications are made with the closest relative of the extinct species, and it helps in the restoration of ecosystem. So what is CRISPR? CRISPR is nothing but refers to clustered, regularly interspaced, short palindromic repeats. It's a powerful genome editing used by scientists and researchers to edit genes with high precision. Initially discovered as a natural defense system in bacteria and archaea against viruses, officially it is used in 2012 in genome editing. Mainly Cas9 endonuclease is used in CRISPR system for genome editing, guided by short RNA sequence to find out and cut specific sections of DNA. After which the DNA is cut, and it enables the scientist to remove, add, or replace any kind of genetic material to that location with the genes of interest. It revolutionized the field of genetic engineering, as it is faster, cheaper, and more accurate than other tools such as zinc finger nucleases, Talon's meganucleases, etc. Biotech companies are currently using this system to edit the genomes of living species to include genes from the extinct species. For instance, the revival of woolly mammoth involves the collection of genes from the remains of mammoth and edit the genome with the closest relative, that is the Asian elephant, including its key traits such as fat storage, thick fur, elongated tusk, etc. Many other projects such as revival of the thylacine, that is known as the Tasmanian tiger, dodo, passenger pigeon, direwolf, etc. are currently in progress. Organizations like Revive and Restore, Colossal Biosciences and Laboratories, etc. are leading these efforts. Their primary goal is to restore the lost ecosystem and boost biodiversity in the world. So what are all the bioethics? Although these recent advancements are curious to talk about, they also raise deep ethical questions between among. The ethics of de-extinction has barely received philosophical treatment. We also provide a novel analysis of the main ethical considerations, social implications, public perception in this review. So next, what is the current mechanism of CRISPR in de-extinction process? So in de-extinction, the process begins with identifying, sequencing the genome of the extinct species and extracting the DNA from the preserved remains of the extinct species. Then the genome sequence is compared with the closely related extinct species. Using CRISPR-Cas9 endonuclease, scientists use guide RNAs that target the homologous regions in the genome. The guide RNAs, which is directed by Cas9 nuclease, induces site-specific double-stranded breaks, and the cell's natural repair mechanisms such as homology-directed repair and non-homologous end-joining are then exploited to insert or substitute genetic traits of the extinct species into the genome of the living proxy. Iterative editing across many loci is necessary to achieve functional restoration of extinct species. After the successful editing, cells are screened for correct modifications using next generation sequencing. You can clearly know what are the details I said from this picture. The DNA is cut by the guide RNA using the Cas9 endonuclease, and the gene that is not necessary for the editing or removed, and the necessary genes from the closest relative are extracted from it and is inserted, forming a new genome sequence, and it's implemented into the egg of a surrogate mother, and the species is revived. So these edited cells are propagated and used in somatic cell nuclear transfer or direct zygote editing to generate embryos carrying the restricted traits. So why Cas9 has been used? CRISPR-associated protein Cas9 has become the preferred molecular tool for de-extinction projects because of its exceptional precision, programmability, and efficiency in genome editing. Unlike earlier genome editing methods such as zinc finger nucleases and transcription activator like effector nucleases, which require protein engineering for each target, Cas9 only requires the simple redesign of guide RNA, significantly reducing the time and cost of genetic modifications. This ease of reprogramming is critical for de-extinction efforts, where numerous genetic edits across multiple loci are necessary to reintroduce lost traits. Cas9 also enables simultaneous multiplexed editing, allowing dozens of genomic sites to be modified in a single experiment. This relatively small size compared to other CRISPR-associated proteins such as Cas12 and Cas13, which also improves delivery into cells using viral vectors, microinjection, or electroporation methods. Cas9's reliance on the protospacer adjacent motif, which is a short DNA sequence, further enhances its targeting precision while minimizing of target cleavage when optimized guide RNAs and engineered Cas9 variants are used. Cas9 is compatible with both non-homologous and gene-joining homology-directed repair pathways, allowing for both gene knockouts and precise gene insertions respectively. So in the case of de-extinction, SDR-mediated insertions are used to integrate ancient DNA sequences at orthologous locations, reconstructing the genetic architecture of extinct species within extant relatives. Thus, Cas9's flexibility, precision, and delivery advantages make it the most effective genome editing system currently available for the de-extinction programs. So why Cas12 is occasionally used and why Cas13 is not used in this de-extinction process? Cas9 remains the most widely used CRISPR-associated nucleus in de-extinction, while Cas12 has seen some occasional use due to the following properties. More like Cas9, Cas12a introduces, also known as CPF1, a double-strand breaks in DNA, but differs mechanistically by generating sticky ends instead of blunt ends, facilitating more efficient homology-directed repair. Cas12 expands the range of editable sites in AT-rich regions of genomes, which is an important consideration when reconstructing large extinct genomes by recognizing T-rich protospacer adjacent motif, which is known as PAM. Cas12a plays a role in simplifying the editary machinery, reducing the construct size, making it potentially easier to deliver into cells. The reason behind it is it requires only a short CRISPR RNA, which is known as the CRRNA, without the need for trans-activating CRRNA, which is known as the tracer RNA. In contrast, the main reason behind Cas13's exclusion is the process of genome extinction, genome editing in de-extinction is it targets RNA and not the DNA. Since the DNA changing genome editing is the main process behind de-extinction, as Cas13 targets the RNA, it is not used in the process of de-extinction. As Cas13 enzymes such as Cas13a and Cas13b.
Kailash, once again.
Yes, sir.
Kailash, make it more precise. What actually you are going to conclude? Just elaborately, you are telling about all the theoretical aspects. So precise with the slide to 10 to 15 seconds in each slide. You're studying all the contents in the slide.
Okay, sir. I'm taking much time in a single slide.
Yes.
Okay, sir. I'll get a little bit fast. So Cas13 is not used as it is mainly used in RNA editing. That's the main reason for why Cas13 is not used. So these are all the comparison table between Cas9, Cas12 and Cas13. Cas9 and Cas12 are used in the double-stranded DNA and targets it, while Cas13 is targeting the single or standard RNA. The cleavage type is blunt ends in Cas9 and sticky ends in Cas12, where in Cas13 it's RNA cleavage. PAM requirement is G-rich, where it is T-rich, and no PAM use, and it uses protospacer flanking site. Guide RNA requires CRRNA, tracer RNA, here only cRNA, and in case of Cas13 is also cRNA. HDR compatibility is worked with mostly with HDR. In case of Cas12, it's also HDR, but with the end of sticky ends. And in Cas13, RNA is transient and is not editable. commonly uses in gene editing, gene knockout, in this case of synthetic biology in Cas9, and Cas12 is genome editing in AT-rich areas, alternative PAM sites, and in Cas13 is RNA interference and diagnostics. So coming to the main topic of the revival of the lost, which is the de-extinction process. So extinction is the colossal problem facing the world, and Colossal is the company, that's known as the Bio-Colossal company, that going to fix it proclaims Colossal Laboratories and Biosciences. So for Colossal, this involves combining the science of genetics with the business of discovery in order to bring back the extinct species, which will not only help revive the lost habitats, but also contribute toward making humanity more human. So these are all the current projects that are working by Colossal Bio Company for de-extinction process. Woolly Mammoth, Doodoo, Direwolf, and Thalassemia. So recently, Colossal Biosciences has successfully done the return of the Direwolf project. So Direwolf is a kind of wolf that has lived across the American range, far south as Venezuela, as far north as Canada, over 10,000 years ago, and were extinct. Plenty of direwolf remains have been discovered across Americas. However, that presented an opportunity for the company. Romulus and Remus were two direwolf pups to walk on Earth after 10,000 years. At young age, they were about 4 feet long, weighing 80 pounds, and possibly go up to 6 feet length and 150 pounds. Relying on deft genetic engineering and ancient preserved DNA, Colossal scientists deciphered the direwolf genome, read the genetic code with the common ancestor grey wolf, matched it with the domestic dogs as surrogate mothers. Both Romulus, Remus, and their sister 2-month-old Kelsey into the world during these 3 separate births, last fall and this winter. Effectively, for the first time, de-extinct a line of beasts whose live gene pool long ago vanished. So this is the picture of the L-Coral gene that you can see in the right side of the slide. So this is the gene that is the main differentiating it from the grey wolf and the direwolf. So actually, the process is done with the rerouting of 14 key genes in the cell's nucleus to match those of the direwolf. No ancient direwolf DNA was actually spliced into the grey wolf's genome. The editing nucleus was then transferred to the denucleated ovum. The scientists produced 45 engineered ova, which were developed into embryos in the lab. Those embryos were inserted in the wombs of 2 surrogate horn mixes, chosen mostly for their overall health and not insignificantly their size, since they were giving birth to large pups. In each mother, 1 embryo took hold and proceeded to a full-term pregnancy. No dogs were experienced a miscarriage or stillbirth. So on October 1st, 2024, the surrogates birthed Romulus and Remus. A few months later, Colossal repeated the procedure of another clutch of embryos and another surrogate mother, and June 30, 2025, that dog gave birth to Kelsey, which is the third direwolf to open it. So from here, my colleague Swati will take.
Good morning to all. I'm Swati. I'm bioethical and social implications. Ecosystem have changed a lot since lots of species. The extinct and so many debates are going on the ethics behind the use of genetic engineering. The says of Robert Klitzman, professor of psychiatry and director of the bioethics master program at Columbia University. There are lots of risk to take. There a risk of death, there's risk of side effects that are severe. There are a lot of suffering involved in that. Challenges and ethical concern. Incomplete Asian DNA. Incomplete Asian DNA is one of the most fundamental challenge in de-extinction. This makes it difficult to assemble a complete and accurate genome from cloning or genome editing. Ethical concerns to recreate life, as the resulting organism may suffer from physical or behavioral abnormalities. Next is surrogate health risk. Extinct species into a closely related to surrogate. they lead to biological incompatibility. This caused by developmental issues, they resulting in miscarriage and premature birth. Impact. Ecological impact, the ecosystem have here changed since this species meaning their reintroduction may disturb current food webs or displace the extinct species. Playing God or restoration of ecosystem. The concern of playing God, the idea that humans should not reverse natural extinct or interference so directly with evaluation. Critics argue the extinction is often a natural process and the reviving species may cross moral boundaries. The welfare of the extinct animal is a major concern. These animals could suffer from health issues, developmental abnormalities, or social isolation, epically if raised without their own kind. The cloning process may recruit multiple failed pregnancies or invasive procedures in living animals, raising concerns about exploitation and suffering. Some argue that de-extinction could shift the public and the scientific focus away from urgent effort to protect endangered species and habits. Public perception of de-extinction. Public perception of de-extinction is largely derived by media narratives and the appeal of restoring lost biodiversity. Species such as the woolly mammoth, dodo, Thailand, passenger penguin, etc. carry symbolic meaning at the
conclude your topic actually. We are going studying all over the slides.
Oh, okay, sir. Ronald Sandler, a philosopher, well known as his work in environmental ethics, not the public reaction, infused by ethical intuition about naturalness or authenticity. Future emerging projects including the extinction of woolly mammoth, select the development of process of the basic of bird propagating mammal and woolly mammoth. The woolly mammoth is a cold resistant.
Conclude your topic. Come to the conclusion.
The conclusion is advantages of CRISPR technology is using in transformation of era for genetic engineering. And the de-extinction challenges and boundaries between natural synthetic life raise concern about animal welfare and ecological destruction, and force society to confront the moral implications of playing God. Moving forward, it's essential that a scientific innovation process alongside transparent public discourse, strict ethical oversight, and multiple disciplinary collaborations. Thank you, sir.
Thank you for your presentation, Kailash and Shweta.
Swati, sir.
Swati. Kailash.
Yes, sir.
Okay, so what you're going to conclude at the end of this review, or you have a plan to do further any research on this review article?
Yes, sir. Actually, we got some less time to do the research, so we can't get more info on that.
Okay. Okay, more graphical representations or the tables will be good. It will be if it is in your presentation.
Yes, sir. Actually, this is currently going on, sir. So there is no such current review paper on this. The company, by Colossal Patients, didn't release any kind of review papers or research paper on this topic, sir.
Okay. Okay, fine. Thank you.
Yes.
Thank you, sir. Shall we go to next presentation? So participants M. Sudarshan, M. Uma Shankar available. Paper on the topic deep learning methods for lung cancer segmentation. Participants available.
I think they are not available, ma'am.
Okay, sir.
You can move on to next
Next
presentation.
participants L. Shruti, N. Srinivasan are for the paper targeted drug delivery. Participants available?
Yes, ma'am.
Okay, you can start presenting. Please share your screen.
இல்ல ரிவ்யூ பேப்பர் இல்ல இது வந்து இதுதான். Is my screen visible?
Yes.
Good morning to one all present here. This is Shruti, Srivatsan and Basmati from Bharat University. We are here to present a review paper on the topic drug delivery system. Here are the contents we are going to discuss about today. This review paper on targeted delivery system is nowadays a highly preferred and facilitating field of medical world. The targeted drug delivery system is a technique used to target the specific side of the body, such as disease cells or organs, while minimizing the exposure to healthy cells. This can be achieved by various approaches such as nanoparticle-based delivery system, monoclonal.
Topic is nanotechnology, enhanced medical devices, current status, and future perspective.
Okay, you can start.
So just a minute, sir. I'll sharing the PPT. So now we are going to see about nanotechnology. Nanotechnology is just as a nano implant, which is the biomedical engineering offering transformative potential in diagnosis, treatment, and management of various medical conditions. This helps to, like, for future conditions. Hello?
It is audible. You can continue.
Yes, sir. Now we are going. This is what's our overview is. We are going to see about the future perspective and current status of the nanotechnology. So this helps to enhance the different types of applications which is required for nowadays. This helps for the drug delivery, improved electrical conductivity, and reduced risk of infection. Yeah. Application of nano implants can the wide spectrum of medical fields, including orthopedic, cardiology, neurology, and oncology. For instance, nanostructures. Orthopedic implants promotes better bone cell additions and faster healing. Nanoplants. Orthopedic nanoplants is just as a nano texture titanium surface, promote better bone cell additions and osseointegration. Nano hydroxyapatite coating mimic natural bone mineral to enhance compactability. For example, it's a best example as Zimmer Biomet's dental implant with nanostructure surface, which is nowadays most widely used in the medical field. Cardiovascular nano implants. It's helped as nano coated stents like anti-inflammatory or anti-proliferate drugs to prevent restenoids. Silver or gold nanoparticles are most widely in this field for antimicrobial protection on pacemaker leads. Example, Cypher and Taxus. Ocular nano implant, which helps us drug deliver implant, and it's a rare case. It's a best example of Diastia, a sustained release drug delivery system for glaucoma. Cancer targeting nano implants. It's a basic implants which is used as an implant coating magnetic or gold nanoparticle that can be externally activated. For example, experimentally implantable nanofiber delivering chemotherapy directly to tumor sites. There's a loss of biosensing nano implants, which help us implantable nanosensor that can continuously measure physiological parameters and transmit data wireless.
The slides are not moving.
Sorry, sir?
Slides are not moving.
Yeah,
just check once.
So there is recently we have research about the drawbacks of the nano implants, which is explained by my friend Nishant.
Hello, sir. My voice is audible, sir?
It's audible.
Yes. Hi, sir. From drawbacks of nano-implantable devices, their main drawbacks are biocompatibility and toxicity concerns. Some nanomaterials, certain metal oxides, or carbon-based nanomaterials may provoke immune response or inflammation. Long-term degradation particles in tissue is not fully understood. Complexity manufacturing and high cost. Advancement equipment. Hello, sir. My voice is audible, sir?
It is audible.
Hi, sir. Fabrication at nanoscale requires advancement, equipment, and strict quality control. Costs are significantly higher than traditional implants, limiting accessibility in lower resource settings. And then regulatory changes, lack of standardization protocols for evaluating the safety and efficiency of nano-based devices, difficulties in gaining regulatory approvals due to novel materials properties and long-term safety uncertainties. And the ethical and privacy issues also there. Nano-enabled biosensors may continuously collect health data. Here, main concern about the data privacy and concerns. Integration with AI and IoT can lead to ethical debates on autonomy and surveillance issues. And then limited long-term clinical data. Nanomaterials, many nano implants are still in early trial phases only. Long-term human studies are also lacking. Unknown interactions with aging issue or concurrent medical treatments also. And then the future impacts and future directions. Precision medicine and smart therapeutics. Tailored treatment through nano implants that adapt to patient real-time psychological signals. Respond to triggers like pH, temperature, and glucose levels of the human condition.
Ultra-sensitive nanoelectrodes may enable better brain computing interference, helping in prosthetic control, paralysis recovery, or even enhancing concentration, and helps to develop new neurons also. Early disease detection. Implantable nanosensors can detect molecular changes before symptoms appear, enabling preventing intervention in disease like cancer or neurodegeneration. Generative medicine integration. Nanoparted implants could guide stem cells and tissue regeneration, improving outcomes in orthopedic and soft tissue implants. And then remote health monitoring. Future nano implants may allow for continuous health monitoring via wireless transmission. This helps to reduce hospital visits and enabling real-time telemedicine. At last conclusion session, nano-implantable devices represent a transformative in modern medicine, offering unpredictable capabilities in diagnostics, treatment, and tissue integration. By operating at a nanoscale, these implants can interact directly with the cellular and molecular processes, enabling smarter or more responsive and highly personalized healthcare solutions. From enhancing biocompatibility and reducing infection risk to enabling real-time health monitoring and targeted drug delivery, the potential benefits are also vast. However, these advancements are not without challenges and limitations. Issues related to biocompatibility, long-term safety, and high production cost and ethical considerations must be truly addressed through interdisciplinary research, standardization regulations, and transparent public dialogue. As technology continues to evolve, nano implants are poised to revolutionize the future of medicine, shifting branding from reactive treatment to proactive precision-based care. That's all. Thank you, sir.
Okay, thank you. So, any future idea on this nano implants for your project or something based on your review?
Yes, sir. By checking the biocompatibility materials and surface coating, enhance the wireless charging efficiency to develop more precise non-implantable devices.
Any sources you have identified for developing this nano implants?
Like what, sir? What kind of sources?
By which you can develop this nano implants?
By nanomaterials, like silver.
Transfer to heart is blocked. Myocardial infarction occurs to damage. This project presents a multimodal network that integrates the ECG signal, echocardium images, and videos and patient data accurate prediction using LSTM and AN transformers and attention mechanism. The model captures key spatial and temporal features. Enabling effective myocardial infarction is a critical condition requires early diagnosis to improve the outcomes. Your ECG is widely used for MI detection, that means myocardial detection. This project proposed the deep learning model combining ANN and LSTM. LSTM captures the temporal patterns in the signals while ANN handles complex features learning. Trained on real clinical data set, the model shows high accuracy and real
myocardial infarction detection. These traditional methods will rely on the clinical assessments and the ECG blood biomarkers. Plus, the traditional methods requires expert interpretation and may also have errors in it. So, we are proposing a fully automated myocardial detection using ANN and LSTM, enhancing accuracy and efficiency. This model is trained on labeled ECG data, enabling real-time MI classification with improved sensitivity and specificity. This system offers a rapid and reliable method for early detection of myocardial infarctions. The working principle of this, we have acquired the data set from Kaggle.com for our project. the first step for the pre-processing. In the pre-processing we have removed the noises and segmented the signals for better
Slide
audio
moving
for
up, Joy.
yeah. 1 second, sir. We'll check it off.
Sir, actually, we are facing some internet issues. We'll resume within seconds.
Okay, okay.
2 seconds.
Participants Aishwarya V. Subashri. Sir, shall we move to next paper presentation?
Yes, ma'am.
Next participants T. Dinesh Kumar and N. Praveen Kumar. Paper title Biosynthesis of Nanoparticles and their Wound Healing.
Good afternoon, sir. I am audible.
You are audible.
Good afternoon, sir.
Good afternoon.
Sir, screen is visible.
Yes, it is visible. Can start now.
Good morning, sir. I am Dinesh Kumar from Muthukumar Institute of Technology. Currently pursuing final year. I am here to explain the biosynthesis of zinc oxide nanoparticle and
Your voice is not audible, Dinesh.
their wound healing properties. The wound healing is audible.
Not clearly audible.
Sir?
Check once. Not clearly audible, your voices.
Sir, now it's clearly audible.
Yes. You can start, Dinesh.
Thank you. This study presents the green synthesis of zinc oxide nanoparticle using the Califa indica plant and the extract and evaluate their wound healing properties. This helps the wound healing property and the antimicrobial activity very strong in this extract aqueous solution and we synthesis with the zinc oxide nanoparticle and confirm the peak wave by UV visible spectroscopy. Then we conclude the antimicrobial activity like antibacterial and antifungal activities against the pathogenic strains, exhibiting the microbial effects. Then we prepare the biofilm using a polyvinyl alcohol and the carrageenan based matrix, which helps the material for the wound healing. First, we use the zinc oxide nanoparticle for their antimicrobial and wound healing properties, and when it is more toxic and energy intensive, then we check the green synthesis for the phytochemical activities. Zinc oxide nanoparticle shows the antimicrobial activities using the bacteria like Staphylococcus aureus and Pseudomonas aureus on nutrient agar medium. For the antifungal activity test, Aspergillus niger and the Candida albicans fungus is used on the potato dextrose. Then we conclude that the antimicrobial activities are good. Then we move to the biofilm preparation by using PVA and carrageenan. This is a collection of plant. We collect the fresh Alkefa indica leaves from the local environment, and then we dry the plant and powder the sample. Then the process of extraction of aqueous solution, preparation of plant material. The fine powder dried alkali indicas are used for the extraction. Aqueous extraction process is the 2 grams of powder material was sweetened and added to the 5 ml of distilled water. Then it was heated at 60 to 80 degree 30 minutes with a continuous stirring. After heating, the solution allowed to cool at room temperature. Then we filter the solution and we get the aqueous excess collected from the flask. Then we test the water solution for the qualitative phytochemical screening with the Alkefa indica extract. This phytochemical tests through the alkaloids, carbonides, glycosides, saponins, proteins and amino acids, phenolic compounds, flavonoids, terpenoids and steroids present or not. Then we move to the green synthesis of zinc oxide nanoparticle. We combine the extract solution 1:1 ratio to the zinc oxide nanoparticle and heat gently and observe the color change. We observe the color change to white color. It indicates the formation of zinc oxide nanoparticle. Then we characterize the nanoparticle presence by using the UV visible spectroscopy. It has the peak waveform of 250 Hz presence of bioactive components that act as reducing agent during nanoparticle synthesis. Then the antimicrobial testing for the zinc oxide nanoparticles. The bacteria uses the Staphylococcus aureus and Pseudomonas aerugis. And for the fungal, we use the Aspergillus niger and Candida albicans in potato dextrosmerium. Then the preparation of biofilm using a PVA garagine with zinc oxide nanoparticle. The 5 ml of polyvinyl alcohol is in the 100 ml of distilled water. Carrageenan we take 1.5% solution. 5 ml of zinc oxide nanoparticle solution.
You can come to what are the results you have done. No need to explain the procedure.
Yes. So it's under testing only, sir. We cell culture is on process, and then we have to conclude the wound healing properties.
Okay, okay, good. My question is why particularly you have gone for a zinc oxide nanoparticle? Other nanoparticles are available. Why you have chosen the zinc oxide nanoparticle? Am I audible?
For the antibacterial activity.
Voice is not clear, Dinesh. Voice is not audible, Dinesh Kumar.
Sir, audible?
Now only it is audible.
Sir, it has antimicrobial properties and anti-inflammatory effects, so that it helps for the wound healing very clearly, sir. So that we use zinc oxide nanoparticles.
Yes, in other
And
nanoparticle
helps
it
to
is not
skin.
available actually.
Sir, it's available, sir. For this, it has a skin regeneration and helps with moisture retention for the skin. So that we use zinc oxide nanoparticles.
Okay, good work.
Thank you, sir.
Thank you. Next presentation.
I think the session is going to be over because two of the participants were not available to present their papers.
Okay, ma'am.
So the session is going to be over, sir.
Thank you for inviting me for sharing this session. I hopefully found
Thank you
the
so much.
presentations are good and up to the mark.
Thank you, sir, for your guidance and support.
Thank you, ma'am.
Thank you all the participants. You can join in afternoon session also. The link will be shared in the group. Thank you all.
பத்மா யூடியூப்ல போயிட்டு லைவ் வருதா செக் பண்ணு, கால் பண்ணு, போய் பாரு. சாரோட நேமே வரல மேம். FTP-ல இருக்கா? ஒய்ஃபைலேயே கனெக்ட் பண்ணிக்கோங்க.
Is the screen visible now? Am I audible now? Am I audible? Sir, is it visible now?
ஐயோ ஏன் டச் ஆயிடுச்சுடா ஹார்டின்லாம் பறக்குதுடா ஒயர் டச் ஆகி
Good afternoon to everyone. So the topic is about challenges in drug development process. Okay, in the drug development process, there are 5 stages generally: discovery, hit generation, lead generation, clinical development, and post registration. So, in the drug discovery, generally it contains to identify the target and the resources. So, first stage we will be identifying for which target we need to find the drug, and then where we are going to get the resources for drug development. So, second process is about the hit generation. So, after identifying the target and the resource, we need to develop the process and we need to test the substances. So, after the development process, we need to conduct the in vitro testing process. So, the third stage is about the lead generation. So, in this we will be conducting in vivo testing. So, the parameters will be analyzed as pharmacokinetic and pharmacodynamic studies. Next, the fourth stage is the clinical development, in which there are 3 phases involved: phase 1, phase 2, and phase 3. Initial phase and median phase and the last phase. The last stage of drug development is about post registration. It is the phase 4, post marketing surveillance. So, in the first stage, discovery, first we are going to identify the target and the resource. So, for identifying the targets, we need to analyze some literature review. So, where the area of interest in terms of drug indications, and what are the targets generally available in the existing research, relevant cellular or the molecular targets is available for our drug development, and whereas the appropriate assays have been conducted or not, whether it is fully developed or it is going to be established newly, that we need to understand, we need to analysis based on the relevant literature. Then we need to identify the patent situation in our target area. Any other earlier available patent are there in our targeted area. So next is about the resource identification. So it is about the potential resources for the novel drugs. So many resources can be identified for the new drug development. Main resources are plants, fungi, bacteria, and also animals. So the resources can be done in a combinational approaches also. Combinatorial chemistry we can do, and then we can go for the structure-based drug designs also. We can go for the drug development process. So for this method for drug discovery, we are using the high throughput screening of the random samples and ethnobotanical approach. That is the traditional use of the national plants or the organisms for particular medicines. So next is the resource identification. So example, some plant is Alphnia institute. So the medicinal plant which has the main vital role in the discovery of the novel drugs nowadays. So it has a significant role as a resource for the particular drug development. So in the research says is between 1981 to 2002, 52% of drugs are approved in the US from the natural product or from the derived form. So there are 26 plant-based drugs are approved during 2000 to 2006, which includes novel molecular-based drugs. So in the future, multi-component therapeutics will be a significant role in the biomedicine sector. So the methods for drug discovery. So there is an ethnobotanical approach in a drug discovery process. So first we need to screen the process. That is, so next is the development of test substance. So in the hit generation, R&D, how it is going to work it out, how much efficiency will be there if it is converted into a drug molecule. And then we need to go for a patent draft. We need to prepare a patent for the particular drug molecule. Raw material supply. So the raw material is based on the any plant materials which can be converted into active compound. So available of raw materials and the excipients. So we need to identify the herbal raw material, whether it is an established market, whether it is contrast cultivation is available for particular active compound. While harvesting is done, period for getting a raw material without any lags. We need to identify these things. So we need to give attention for the continuous availability of the raw material, and then quality variations must be there, and the biodiversity regulation should be followed for the raw material, plant substances. Existing patent and IPR should be verified before applying the raw material, before getting in the raw material. So next is we need to identify the raw materials, that is the active compound. So we need to refer the monographs in pharmacopoeia for the identifying chemical substances and the herbal raw material. So what are the contents will be there in a monograph? What are things we need to analyze with the raw material? Chemical characterization we need to do, and then appearance and solubility test we need to perform with the raw material, and then we need to identify with the microscopic techniques or the physiochemical test. So we need to perform qualitative analysis and also the quantitative analysis, and then we need to identify the impurities level also in the raw material substance. So next is the quality control and production. What are the methods for identifying the phytocompounds? So we can identify the phytocompounds using TLC and also by HPLC method. So after analyzing all the data from the module 1, we need to compile it in as an CTD documentation, that is common technical document, so which has to be compiled. So next is the module 3. It is based on the quality of the particular sample, their monograph, their specification, and the development report, how we are going to develop the active compound into a substance, drug substance. So next is preclinical testing. So as said before, we will be doing in vitro profiling in the stage 2. It is biochemical assays, cell culture assays, and isolated tissue assays. So the biochemical is enzymatic activity assay can be performed. In cell culture, we can do various cancer cell lines. Isolated tissue assays, we can perform the mucosa model. So we need to test the drug substance with the toxicological studies also, how much the toxic effects are there, whether it is in the prescribed limit with the plant substance. So we need to identify the toxic effects of the particular plant sample. And then bacterial cell cultures, by this also we can identify the toxic studies. So in the in vivo testing, what are the things to be done? What are the things will be monitored in the in vivo testing? That is it is the lead generation. After completing the second stage, the drug development will be move on to the next stage, lead generation. So in this, the drug action will be tested, and also the toxicology studies will be done. So for in vivo testing, we can use the animal model. So for drug action, the animal's behavior and reaction will be monitored, physiological parameters will be monitored, and then histopathological parameters will be monitored in the animal model. Then toxicity level will be obtained, or it will be identified with the animal models. With this, acute is a small level of toxicity level. Subchronic is the before the chronic stage. Medium stage will be identified, how much toxicity is there, and tissue specific and tolerability also will be checked with the animal models. So we need to get an ethical concern for the number of species or the number of animals used for this study purpose. So in the third lead generation, next stage is preclinical development. So after identifying with the animal models, we need to perform two studies to identify the drug molecule, pharmacokinetic studies and pharmacodynamic studies. So pharmacokinetics is nothing but what does the body to the drug. So it is done by ADMET studies generally, absorption, distribution, metabolism and excretion. Whereas in pharmacodynamic, physiological effects will be monitored, drug action will be monitored, and then relationship between the drug concentration and the effects will be identified. So it is an important parameter in a drug development process, both the parameter pharmacodynamic and pharmacokinetic. So after this process, we need to compile the document, we need to create a database, and it will be sent to the patent process. So for developing a patent strategy, we need to identify the rationale and the possibilities for getting our patent to be granted, and decide strength, what is the strength based on this research for developing a drug molecule, and then cost effective, how much it is cost effective, we need to identify. So next is quality control and production. So scaling up from lab scale to production size, we need to perform the experiment, how much it will be cost from lab scale as such we are doing in lab. If it is increased to large scale production, what are the requirements will be made and what are the things to be done, that has to be verified. So we need to conduct a validation. So it is a process for validating, including various batch sizes which will be performed. Stability testing will be done. So it is a stability test under different conditions of temperature, humidity, time, exposure time, all it will be checked in the stability testing. So after completing these parameters, it will be compiled in a document. So It contains the validation report, stability report, manufacturing protocol, and the development report. So all these modules are called as the quality. So it is called as module 3. So then it will be sent to the next stage of the drug development process. So next is fourth stage, clinical development. As said, it is 3 stages: phase 1, phase 2, and phase 3. So it is the linking bench to the bedside. So from initial stage to the final stage. So this clinical trial will be done with the humans basically. So whereas in a lead stage, it will be done with the animal models. So in this, we will be using human volunteers. So we need to get the ethical concern for conducting this human subjects. So it is mainly done to qualify to increase the existing knowledge. It respects the freedom of decision of volunteer who is involved in the drug development process, and it will be analyzed for the risk benefit assessment mainly. So in the first stages about phase 1 studies, it is conducted for 20 to 30 healthy volunteers. What are the parameters will be monitored? Safety and tolerability, pharmacokinetics parameter, ADMAT, and then pharmacodynamics. So these will be monitored in the phase 1 studies. So based on the phase 1 study reports, if it is satisfied, then only it will be proceeded. Then only it will be approved by FDA to proceed with the phase 2. So in the phase 2, 100 to 500 patients will be involved in the phase 2 studies. So in this, they will be identifying the safety and tolerability, and then same pharmacokinetic and dynamics will be done for more number of patients, volunteers, and then efficiency, how much efficiency the drug molecule is, will be identified. Then the dosage will be fixed in the phase 2 only. Dosage comparison will be done in this phase 2. So the phase 3 it is about more than 1,000 patients will be involved in the phase 3. It is the main stage. After completing phase 1 and phase 2, the documents will be compiled and it will be sent to FDA organization. They will go through all our results. They'll track the ups and downs of the report. Then they will give suggestions. Based on the suggestions, if it is approved, we can go for the phase 3 studies. So it is monitored the reaction for the long-term drug use. If the patient is using in a long-term process, what will be the effects will be there. So it will be monitored mainly in the phase 2. So in this, what will be checked, what will be identified in the phase 3 is comparison to placebo or to standard therapy. So what is the standard protocol for taking this, consuming this drug? Multi-center and multinational trials will be conducted in phase 3. So combination of drugs will be taken into consideration. What are the effects are arising in this phase 3? So overall, it is called as phase 3 is called as risk-benefit evaluation. So next is 2 parameters are there in a 4th stage: clinical samples, production, and the quality control. So in the production, they will provide appropriate sample quantities from the phase 1, phase 2, and phase 3, and then what will be the sample shipment logistics will be there. Whereas in a quality control, we will be having a batch release documentation process. So each and every parameter will be monitored and it will be documented as a batch release documentation. So how the short-term and long-term storage of samples will be defined, it will be defined in a quality control. So all this process will be conducted based on the guidelines of good manufacturing practice and good laboratory practice. So all these documents will be compiled. It is called as the last module is called as clinical study reports. So all the 5 modules will be sent to the regulatory authorities for the approval of a drug development. So next is about after the drug molecule has been approved. The phase 4 is called as the post-marketing testing. So after the drug approved, it will become into the marketing after production. So after it is reaching the consumer, the different parameters will be evaluated. First one is expanded benefit risk profile, and then combination with other drugs, then dosage, optimization of dosage and the uses. So it will be monitored. If any issues arises in this, then the drug will be taken back with their specified batch number. So some issues, if any issues arises, so it will be considered and it will be taken back by the industry agencies. So next is all these manufacturing protocol also will be followed by GMP and GLP guidelines. So once the approval is done by the marketing authorities. The production will start by the companies. Thank you. Any doubts?
லிங்க் படி அந்த ஆதார் நேம் படி அவங்க உள்ள இருக்காங்க. லிங்க் அனுப்பிச்சிட்டு அவங்க ஜாயின் பண்ணிடுவாங்க. லாஸ்ட்டாக போகும். ஆன்லைனில் பண்ணுறதுக்கு எதுவும் ஆன்லைனில் பண்ணுறது எதுவும் அனுப்பிடுவோம். ஆன்லைன் பண்ணுறது தான் பண்ணுறேன். நான் இன்னும் பண்ணேன். ஒன்றும் ப்ராப்ளம் இல்லை. அதெல்லாம் ஒன்றும் ப்ராப்ளம் இல்லை. 2 பர்சன்டேஜ் 2 பேட்சுக்கு 2 பேர் இருக்காங்க. 5 ஸ்டாஃப் இருக்கிறாங்க. அப்படின்னா இருக்கு. ஏன் வேணா கண்டிப்பாக நான் கேட்க பண்ணி கவலைப்படாது. சரி. My approval is actually not that right now, but I'm not including any tax effects in it. I have not blocked the account, but the tax effects are blocked from the tax. I know the reason, please. Yes, yes. Yes, I've got it. But it's not blocked from the tax side. Okay. Padma Devasulu. இங்க ஒரு லேப் விசிட் பண்ணணுமா லேப் சரி சார் நான் எல்லாமே ஓப்பன் பண்ணிடுறேன். ஓகே. பண்ணிட்டேன் நான். அப்சாக்ட் நான் உங்களோட இதில் ஜாயின் பண்ணிட்டேன். பசங்க யாரும் என்னை கான்டாக்ட் பண்ணி இந்த மாதிரி இருக்கு பர்சன்டேஜ் அப்ளை பண்ண எனக்கு கண்டாலும் நான் இங்கே இதே மாதிரி தான் ஐடியும் இல்லை பசங்க ஐடியும் வரல இவங்களும் வரல ஐடியும் வரல பேப்பர் கொடுத்து போனவன் தான் நான் ஐடி நான் வேலைக்கு முடிச்சு வரேன். சரியா வேலை இப்பதான் சொன்னாலும் பேசிட்டு அவர்கிட்ட சொல்லிட்டு வந்து தேங்க்ஸ் அதான் சார் ஃபோன் பண்ண அங்கே தான் ஃபோன் பண்ணுறாரு அங்கேருந்து ஃபோன் பண்ணேன் தான் அதான் எதுக்கு வச்சீங்கண்ணா நீ பார்த்தீங்கன்னா என்ன சொல்லி மனப்பாடம் அது சொன்னேன் சார் இதாக வெயிட் பண்ணுறேன் சார் வெயிட் பண்ணி போகிறேன் சார் நான் அந்த ஹெல்த் சாப்பிட பார்க்குறேன் சொன்னேன் அவரே வச்சுருவாரு ஏன் நிற்க மாட்டேங்கிறாரு ஃபோன் பண்ணி பார்த்தேன் எதுவும் சொல்கிறேன் புரியுது என்ன பண்ணா நான் சொல்லிட்டேன் உங்களை காலையில போட்டுக்க வேண்டியதுன்னு அவங்க மாற்றிக்கிறாங்க யோசிக்க சார் நான் அப்படியா நீங்க வேணாம்
நான் எதுவுமே எடுத்துக்க மாட்டேன் சார். ஏன்னா இதுல வந்து ஃபுல் ஃபிளட்ஜ் வந்து நீங்க தான் ஒர்க் மாட்டீங்க. எனக்கு ஃபர்ஸ்ட்ல போய் எக்ஸ்ட்ரா இருக்குன்னு வச்சேன். அவன் ஃப்ரெண்ட்ஸா வந்து இன்டர் பண்ணுறாங்க. யாரும் பேசல சீக்கிரம் முடிச்சுட்டு ஃபோன் பண்ணுறாங்க. இல்ல சார் அவங்க தான் என்ன பண்ணா பேரு எங்க ஆளே காணும் நான் இப்படி ஏதாவது சொன்னீங்க, நான் இப்படி ஏதாவது பண்ணுவேன். இப்படி பண்ண மாட்டீங்களா? நான் கண்டிப்பா இப்படிலாம் வந்து யாரையும் வந்து கஷ்டப்படுத்தி நம்ம நல்லா லீவ் ஒன்று சொன்னா அது நான் வந்து எப்படின்னா வந்து நான் வரேன் ஏன் சார் அப்போ என்ன அவர் எப்படி சொன்னா சார் அவன் சொல்றாரு தான் சார் அட்மிஷன் ஆளு நான் மட்டும் டெய்லி வரேன் சார் அப்படின்னு சொன்னாங்க. எனக்கு ஒரு ஸ்டாக் அதை மட்டும் தான் தெரிஞ்சுதான் இப்படி பார்த்துட்டு தான் இருக்கும். அதனால பின்னால கேட்டேன். இப்போ இது வச்சுக்கேன் நான் கிணறுல தான் கேட்டேன். தேவை இருப்பின்னு வர சொல்லலாம். தேவை கண்டிப்பா நான் அவங்கதான் கேட்டாரு. அதான் நான் கண்டிப்பா நான் பண்ணினே அவங்க கேட்டாங்க. அதனால நான் அப்படியே டேரக்டா உங்கள்கிட்ட ஏன்னா எனக்கு எதுனா நான் அவங்க கேட்டு சொல்லுவேன். அதனால அவர் தான் சொல்லி அவர்கிட்ட கேட்டு பை பை. எல்லாரும் பிரச்சனை பண்ணதுக்கு ஆமா அப்படின்னா நானும் எங்க போய் லிங்க் லிங்க் போட்டு வீட்டுக்கு போகும்போது அப்படியே உள்ள கவுண்டிங் மாதிரி சார் முடிஞ்சு சார் கொடுத்துலாம் இல்லை சார் அம்மாட்டு நானா அது பசங்க அவன் அவனுக்குள்ளே பேசிக்கிறான். எருமை பக்கத்துல தானே நான் நின்று இருந்தேன். அத்தமாவே பேச மாட்டான். இல்லைன்னா சங்கீதத்துல அப்படியே வீட்டுல பாட வச்சு அவரே ப்ராஜெக்ட் பண்ண வச்சு
இது ஒரு டாப் கிளாஸ் எடுத்தோம். உங்களோட நம்பிக்கை என்ன தான் காலேஜ் முடிஞ்சு நீங்க பேப்பர் அனுப்பிச்சிட்டீங்கன்னா ஃபுல் பேப்பர் இருக்கு ரெடி பண்றேன் அது அப்படியே நீங்க பேப்பரா அனுப்பிச்சிட்டீங்கன்னா பேப்பராகவே கம்யூனிகேட் பண்ணிருந்தேன் எதுக்கு அப்ளை படிச்சு நம்ம முன்னாடி ரொம்ப நான் கம்யூனிகேட்
கம்மியா இருக்கு சார்.
அடுத்த வருஷம் கம்மிஷன் பரவாயில்ல நான் சொன்னேன் உங்களுக்கு தான் தெரியும்.
தேங்க்யூ சொல்லிட்டீங்களா?
Good evening to everyone. I have taken the session to thank all my students, participants, and faculty members who have contributed their entire hard work to complete this first international conference on medical devices and drug delivery systems on this wonderful evaluatory session. In this moment, I would like to thank all the participants from various institutions who have Autism by Nandana, Sharmila S, Shobana P. Sudha from PSNA College of Engineering. The earlier said smart nano capsules by Hadia Begum, and the best innovative innovation system for individuals with autism. Sharmila, Shobana, and P. Sudha. Sharmila, Shobana, and so these are the best presentations and best innovative awards we are declaring.